Good evening, everyone. Welcome to this week's talk with Emily Flashman, who will be giving a talk titled Coming Up for Air, How Plants Sense and Respond to Flooding. Emily did her undergraduate degree at the University of Southampton, then did her DPhil with Professor Chris Schofield at the University of Oxford. She is now a research fellow in enzymology who is primarily focused on studying enzymes involved in oxygen sensitivity responses, both in plants and animals. If you have any questions for Emily during the talk, please write them in the comment section below and we will read them out afterwards. Emily, thank you so much for taking the time to join us. I look forward to hearing your talk. Over to you.
And thank you very much for the invitation to give this talk. It's a real privilege to come and talk to you all about the work that I do. So as Anka said, I work in the Department of Chemistry, and I consider myself a biological chemist, specifically an enzymologist. So I work on enzymes, and enzymes are biological molecules, proteins that catalyze chemical reactions. And we're specifically interested in some enzymes which catalyze cysteine oxidation. And I'm going to talk to you today about how cysteine oxidase enzymes in plants are involved in sensing and responding to flooding events. Okay, so this is not just an instruction for me to calm down at the beginning of a nerve-wracking talk, but it's a reminder for me to talk about how all aerobic organisms need oxygen. And we need oxygen because we, through the process of respiration and metabolism, we convert oxygen and glucose to form ATP, which is the unit of energy that our bodies use. Now, we as humans and animals have respiratory and circulatory systems that enable us to deliver oxygen into the cells of our body. But plants don't have these. They just have to get their oxygen by diffusion. So how do plants breathe? Well, you're probably well aware that plants can photosynthesize. So they can produce oxygen by combining carbon dioxide and water with sunlight to make oxygen and glucose. So that's one source of oxygen, but that only takes place in green leaves. But they can also acquire oxygen via diffusion, both through stomata, which are pores on the leaf surface, but also diffusion through the roots via their root hairs. So this is how plants get their oxygen, and the oxygen diffuses through the plant. But what happens if the plant starts to become waterlogged and flooded? Well, if that happens, then gradually the plants lose their ability to take on oxygen. And as floodwaters increase, the opportunities, the available space for oxygen to diffuse into the plant is reduced. And if the plant is completely submerged, then photosynthesis stops because as well as carbon dioxide not being able to diffuse into the plant, the sunlight is reduced because floodwaters are very murky. Now, obviously, without any oxygen present, then this aerobic metabolism process can't take place. And so this efficient method for producing lots and lots of this ATP, the energy molecule, is removed. And without the oxygen, that can't happen. So instead, what plants do is they switch to an anaerobic metabolism where by a different metabolic route, glucose is converted into carbon dioxide and ethanol, and each glucose molecule forms 2 ATP molecules as opposed to the 38 ATP molecules that you get with aerobic metabolism. So it's a much more inefficient process, and if plants become flooded, that means that they have to, in order to maintain an energy supply for survival, they have to use up lots of glucose. So essentially, they have to survive on their carbohydrate provision until the floodwaters subside. And for many plants, that that doesn't, the floodwaters don't subside quickly enough, and so they succumb to cell death because there's not enough energy provision, but also because when you convert to this anaerobic respiration process, it sounds a bit what's the word I'm looking for, it doesn't quite make sense on the face of it, but you get an increase in reactive oxygen species because you're not coupling oxygen to oxidative phosphorylation properly. So this increase in reactive oxygen species also causes a lot of damage to the protein and DNA and lipids in cells, leading to this type of cell death and crop death. Now, there are many parts of the world where plants are regularly exposed to flooding events. So plants that live on floodplains have gradually adapted through evolution to have traits that allow them to survive being flooded. And this picture gives some examples. So here we have rice. We have rice that have come from this is a lowland variety of rice, and this is a variety of rice that's able to survive deep water flooding. And the difference between these two types of rice is it's called elongation. So the sort of difference in the length between where these different we'll call them petioles come out. So the different branches coming off the stem, the length between where these different branches come off the stem is longer in the rice plant that's better adapted to survive being flooded. So it reaches above the level of the flood water, enabling oxygen to enter the plant at the top of the plant. We see a similar response in Arabidopsis here. So Arabidopsis that is better adapted to flooding has got a greater length of this petiole. So that's the distance between the leaf and the stem, because this has grown to reach above the level of the flood water. Similar over here, this is a sort of marshy plant here, and this form of this plant here is well adapted to flooding. This shows shoot elongation happening so that the plant can grow rapidly to reach above the level of the flood water. So this survives in marshes. Some plants have a very sort of waxy surface on their leaves, which allows a gaseous layer to exist on the surface of the leaf to allow gas exchange even when there's when they're submerged. And then there's some adaptations in roots as well. So adventitious roots are roots that grow above the level of the soil effectively. So you can see here the roots sprouting out from above what would normally be the soil level. And what this does is it means if these adventitious roots develop above the level of the flood water, or this is particularly the case when you've just got soil waterlogging, then these roots are not surrounded by water. They're exposed to air, and oxygen can diffuse in through these roots. Another adaptation that's very important is the formation of erenchyma. And erenchyma are basically big holes in the plant tissue that stretch along the stem. So, for example, all the way up here, you would have erenchyma formation down into the roots as well. And what that does is it enables oxygen that has got into the plant via any tissue that's escaped above the level of the flood water to travel down the plant through the erenchyma. So it facilitates diffusion of the oxygen that's got into the rest of the plant. And then one final adaptation is to prevent diffusion of oxygen out of the roots. So we've got, oh gosh, sorry, excuse me a minute. Just getting rid of the pet. Oh my, can you get rid of the dog? Right, sorry, height of professionalism. Okay, so you, where were we? Yeah, so the roots. So you've got these parenchyma forming down through the shoots. So oxygen is diffusing through the plant into the roots. So what would be a real loss would be if then oxygen diffused out of the roots as it got into the roots and didn't manage to get all the way down to the right to the bottom of the roots. So you get a sort of hardening of the surface of the roots around these sorts of zones so that oxygen can't diffuse out. So that's formation of what's called a radial oxygen loss barrier. And I've got a few more examples of adaptations to flooding, and these pictures were taken by someone in the flooding field when they went on a trip to the Peruvian Amazon, and it just shows some nice adaptations. So this one here shows a special type of fig tree which grows roots which sort of exist above the level of the floodwater, so that as the floodwater goes up and down over different seasons, the roots are exposed and allow oxygen to get into the tree. I'll come back to this one in a minute. But here again, we have an example in this water-adapted hyacinth of petioles that have extended, so very extended petioles. So the distance between the leaf here and the stem is extended so that the leaf can get above the level of the floodwater. Here again, we have this internode elongation. So the distance between the formation of different leaves coming off the stem is very long. The plant is growing rapidly above the level of the floodwater. So most of the mechanisms that I've talked about so far are escape mechanisms, where the plant is growing rapidly above the level of the floodwater and is developing adaptations to allow oxygen into the plant once it has escaped out of the floodwater. But another mechanism, which I alluded to earlier, is a sort of quiescent mechanism. So their plant goes into a metabolic shutdown. It converts from aerobic to anaerobic metabolism, and it tries to sit out the flood until the flood subsides, and then comes back to life. Growth restarts again. And here we can see a sort of gradient of vegetation that demonstrates how this happens. So you've got different layers here where you can see how the floodwater goes up and then comes down, and you can see different extensive vegetation growing that have been initiated since the floodwater has gone down. So here you've got extensive vegetation, here you've got less extensive vegetation, and here you've got very little, because the floodwater has only just subsided from this region. Okay, so all these adaptations have taken place in plants that live in environments where they're so they have adapted over the course of evolution to live in environments where flooding can occur. So they've developed these adaptation mechanisms. And of course, this has happened over a long period of time, but these days we're beginning to experience differences or rather increases in the frequency and intensity of flooding events. And so plants and crops are being exposed to floods without the ability to adapt to flood situations, and they're not dealing with it very well. So crops that we typically grow for food generally cannot survive submergence or flooding for long. They will go into the anaerobic response, and they will use up their carbohydrate reserves, and they will have oxidative damage, and they will die. So I'm sure you can imagine plenty of scenarios that you've seen over the news over the last several years, but this is an example of a flooded banana crop in Mozambique. This is an example of a flooded rice crop in China. This is an example of fairly recent flood-induced death in maize crops in the US. And more locally, in Oxford, allotments have regularly flooded over the past decade or two. And indeed, I used to be an allotment owner, and when I had an allotment, there was one summer where there were severe floods, and all my produce was damaged, and it was heartbreaking. And that's just a small scale allotment. That's just me. So as climate change comes upon us, we can see that there's an increase in flooding. So this is just over the past, well, I mean, this finishes at 2009. So this is the 6 decades leading up to 2009. And it shows that the increase in major flood events in different areas of the globe. And you can see that in particular in Asia, there's been a massive increase in the number of flood events. And then this map shows predicted future flood events with showing how often what have till now been considered 100-year event floods. This predicts how often they are thought they will return in future. So the darker blue indicates the frequency with which major floods will with which these regions will be exposed to major floods. So we can see particular area, dark blue areas again in Southeast Asia, in Central Africa, and South America, and we're not immune to that ourselves in Northern Europe. So flooding is an increasing problem, and plant adaptation is by natural evolution is not going to keep up. And so if we want to be able to feed the 9 or 10 billion people that are expected to inhabit Earth by the middle of this century, then we are going to have to find ways to make sure that the crops that we grow have resilience to these flooding events. Otherwise, we will have a major food security problem. So we need another green revolution, and this is arguably underway already. So many scientists around the world are looking for ways to help plants become more climate resilient. So they're trying to find ways to make plants more drought tolerant, heat tolerant, salinity tolerant, and of course flood tolerant. There are, I guess you could classify these in these strategies into two divisions. So one would be sort of classical breeding, so trying to find traits in different varieties of crops that enable these aspects of climate resilience. Typically, these traits might have been bred out for improved productivity over the past 50 years, but if we can identify traits and breed them back in, then that might help crops survive different climates. But also genetic engineering, and so identifying the pathways that underpin these adaptive mechanisms and learning from these and manipulating them could be another effective mechanism. And both of these hand in hand will hopefully come together to bring a suite of solutions for crop growth in the coming decades. So I'm going to talk to you now about the part that we hope to play in this. But first of all, I'll give you an example of a breeding strategy that's really paid off in terms of flood tolerance. So there is a variety of rice that was identified that has particularly good tolerance to flooding. So it was known to be able to survive being completely submerged for 14 days. And from this rare variety of rice, it was identified that the gene locus that was responsible for this flood tolerance is at a position called sub1. So the sub1 gene was identified, and the sub1 gene was then bred into varieties of rice that are much more commonly grown in Southeast Asia. And what you can see here is those varieties where we've got sub-1 compared to, let me find a control, IR64. So here's IR64 without the sub-1 gene, and here's IR64 with the sub-1 gene. And let's find another one. So Samba over here and Samba over here. Here's Samba with the sub-1 gene, and here's Samba without the sub-1 gene. So quite clearly, the rice varieties which have had the sub-1 gene cloned into them are much more resilient to being flooded. So yeah, I didn't actually explain the crucial point, which that this is a flood tolerance field with different types of rice being exposed to flooding and then desubmergence, and then you see what survives. So these varieties of rice have been developed and distributed by the International Rice Research Institute, which is based in the Philippines. And now these types of rice are being widely grown across Southeast Asia, and it's reported that they result in a 47% increase in rice yield in a flood year, but without any yield penalty in a non-flood year. So this is a real success story for breeding. Okay. Now, you could argue that you have to be a little bit lucky to be able to identify a climate resilient trait in a natural wild variety of a crop, and then to be able to identify the gene responsible for that trait and to be able to breed that into other varieties of your plant. So that situation that I've just described with sub-1 being bred into different rice varieties, that's not going to be applicable to all crops. It's just that there was that rice variety that was particularly tolerant that they could exploit. So we turn instead to the possibility of understanding the molecular pathways behind this tolerance and being able to engineer those pathways as another route to generating flood-tolerant crops. So to be able to do that, you need to understand the molecular mechanisms involved. And these were really deduced in plants about, well, over the past 10 or 15 years. And it really started with the rice story, so that the identification of the sub-1 locus really helped piece together what the flood tolerance molecular mechanism is. So here we have our plant, and it's surviving quite happily in air, but when it becomes submerged, the diffusion of gases is reduced. This leads to a buildup of ethylene in the plant, because plants continuously produce ethylene, but it just diffuses out of the plant. But if the plant is surrounded by water, the ethylene can't diffuse out as readily in water as it can as air. So this ethylene acts as a signal, and it acts as a signal to accumulate certain transcription factors. And we'll come on to that in a minute, but in rice, you see accumulation of two different families of transcription factors. We have the sub family of transcription factors, and they inhibit gibberellin production, and that prevents elongation. So without the gibberellin, there's no growth, and that triggers the plant to go into this metabolic shutdown. It's not growing. We also get induction of genes which enable the plant to switch from aerobic to anaerobic metabolism. Over here, however, we have, so in different varieties of rice, which show different responses depending on what type of flood they're adapted to. So this type of rice is adapted to shallow floods, and that might have a long duration. So this type of rice, as we've seen in some of the photos that I showed you before, grows rapidly above the level of the floodwater. And this response is driven by transcription factors called SK or snorkel. And snorkel describes the erenchyma that I described. So these sort of gappy formations along the stem, they're essentially forming snorkels from the top of the plant, where air can get in all the way to the bottom, with oxygen able to diffuse to the plant tissue from the erenchyma within the plant itself. So we've got transcription factors driving these two different responses, the elongation response and the quiescence response. And the snorkel transcription factors do this by turning gibberellin production on. So gibberellin drives growth, and that's what drives the growth to elongate out of the above the level of the floodwater. Okay, so we've got accumulation of transcription factors. And so transcription factors are proteins that promote expression of certain genes. So these transcription factors will promote the expression of genes that drive the adaptive response in the plant. And the clue to the flood, the connection to the flood adaptive response really came from looking at the sequence of these transcription factors. And so there's different examples of these transcription factors in rice, so from the SK and the subfamily, and also their equivalents in Arabidopsis. And Arabidopsis is the plant, the model plant organism used in plant lab work, and it's called sailcress. It's basically a weed. And what was noticed about these transcription factors is that when you looked at the sequence of these transcription factors, first of all, they're very similar when you compare all these different transcription factors, and I'll call them Pr0f transcription factors. They're group 7 ethylene response factors. So they're very similar, but crucially, all of them, except one, the relevance of which is questionable, but can come back to that. All of them have an M and a C at their N terminus. So the N terminus is the start of a protein. So if you imagine you've got a protein molecule and you've got two residues, a methionine and a cysteine. Now, when proteins get made in the cell by the ribosome, many of them will have their initial methionine cleaved off, so removed at the point of protein production. And if you've got a cysteine at the second position, then this methionine is usually removed. So that means that effectively at the N terminus, once the methionine here has been removed, you've got a cysteine residue represented by C here at the N terminus of all of these transcription factors. So why is this important? Okay, now there's a pathway or a certain phenomenon that describes that the identity of the amino acid at the N terminus of a protein will have an effect on the stability of that protein. And that's because having certain amino acids at the N terminus of a protein will destabilize it. It'll send it to what's called the proteasome here, which is effectively the cellular dustbin. So this pathway is called the Endecon pathway. And some of the residues that are destabilizing are indicated here. So cysteine here is not destabilizing by itself. However, if cysteine gets oxidized, then that oxidized form of cysteine becomes a substrate. So it gets recognized by a different enzyme, which adds an arginine to make a new N terminus. And that arginine is a signal for the next step in the process, which sends the protein for degradation by the proteasome. So what we have then is a connection between cysteine, oxygen, and degradation of the protein. So if we think then back to our Pr0f transcription factor, if we've got a high oxygen environment, which we would do when a plant is not flooded. Then we have the cysteine at the end terminus of our transcription factor that gets oxidized, and that oxidized cysteine has an arginine attached to it, and that gets sent for degradation by the proteasome. So the ERF transcription factor is destabilized and does nothing. If you have a reduced oxygen environment, as you do when your plant is flooded, because oxygen can't diffuse into the plant, then that cysteine oxidation does not take place, and your transcription factor is stabilized to induce the expression of genes which allow the plant's adaptive response to being flooded. So we have a connection between oxygen availability and the molecular response. Now, I'm just going to sidestep for a minute to highlight the similarities between this system, where we've got regulation of the stability of a transcription factor by the availability of oxygen, and a equivalent system in humans. In humans, there's a transcription factor called HIF, hypoxia-inducible factor, and HIF is stable in low oxygen conditions to induce the expression of genes which allow us to respond to low oxygen, and those would be genes that lead to the production of red blood cells and the formation of blood vessels. But in normal oxygen conditions, HIF is also oxidized, this time at a proline residue, and that's a signal for HIF to get sent to the proteasome, the cellular dustbin. And so it's not, HIF is not acting under normal conditions. And this is a really important pathway that has been studied extensively in Oxford and beyond, and the discovery of this pathway was awarded the Nobel Prize in Physiology and Medicine in 2019, including Peter Ratcliffe from Oxford. The important point that I want to highlight to you from the human system is that the oxidation event of the HIF, so where you've got the proline is oxidized to a hydroxyproline, this oxidation event is catalyzed by an enzyme, and it's catalyzed by an enzyme called HIF hydroxylase. And that incorporates, so HIF hydroxylase catalyzes the incorporation of molecular oxygen into the proline, and it's that event which sends the HIF for degradation. And HIF hydroxylases are very oxygen sensitive enzymes, allowing a graded response from high to low oxygen. So when a few years after this pathway was identified, a set of enzymes were identified which were called the Plant Cysteine Oxidases, and they were proposed to catalyze the oxidation of cysteine to oxidized cysteine. That really made a lot of sense, because having enzymes that control this important oxidation event is a way of regulating this whole pathway. So these enzymes, these enzymes were discovered by the group of Francesco Licausi, who's since become a close collaborator and has also recently moved to Oxford to work in the Department of Plant Sciences. So this is where we come in. So my group has had a long-standing interest in oxygen sensing enzymes, and so when these enzymes were identified, we were very keen to look at them and determine whether they could play a part in regulating the plant's response to low oxygen and flooding. So I just check the time, see how much detail I've got time to go into. So there's a bit of detailed chemistry here to show the work that we did in our group, which validated that these plant cysteine oxidases were indeed catalyzing the cysteine oxidation. So first of all, we used mass spectrometry with oxygen and heavy oxygen to demonstrate that the enzymes incorporated molecular oxygen from the atmosphere into the oxidation of the cysteine, which they do. So they are dioxygenases using molecular oxygen. We showed that they were indeed incorporating this molecular oxygen into the N-terminal cysteine of the of peptides representing the Pr0f transcription factor substrates. And we showed that the product of the PCO catalyzed reaction is cysteine sulfonic acid. And then importantly, we teamed up with some collaborators from the Institute of Plant Biochemistry in Halle in Germany, who were working on the arginine transferase enzyme to show that the next step in sending the proteins for degradation, which is the addition of an arginine to the N-terminus, we showed that that was only possible with a cysteine initiating peptide, that the arginylation, represented here by incorporation of radioactive arginine, was only possible in the presence of the PCO enzymes. So these N-terminal cysteine had to be oxidized before it could be arginylated. And we demonstrated that with mass spectrometry back in Oxford as well. The next thing we showed was that the PCO enzymes were oxygen sensitive. So here we have kinetic experiments carried out with the 5 different PCO enzymes that you find in Arabidopsis. And what we're doing here is we're measuring the rate of enzyme activity as a function of oxygen concentration. And what we found is that the rate of enzyme activity for all the enzymes was dependent upon oxygen concentration. But not only that, the sensitivity to oxygen concentration was to a point that corresponds with the types of oxygen concentration that you find in plant cells and tissues. So what do I mean by that? Let's take this example over here. So if we compare PCO5 and PCO4. For PCO5, we have, let's call it less sensitive, it's less sensitive to oxygen compared to PCO4, which is very sensitive to oxygen. And what I'm looking at is the gradient of the slope here. So for PCO4, which is very sensitive to oxygen availability, if the oxygen availability dropped from 20% to 10%, then you see a significant drop in the enzyme activity. So if you see a drop in enzyme activity, you're going to see a stabilization of the Pr0f transcription factor because the enzyme's no longer working to send it to the cellular dustbin. PCO is a bit less sensitive. If you drop from 20 to 10% oxygen, you see less of a dramatic effect. But what we do see here is amongst the 5 different enzymes, we see a range of different sensitivities, but all of them are within physiologically relevant oxygen concentrations, which means that if you have deviations from these oxygen concentrations seen in cellular tissues and cells, then you're going to get changes changes in the activity of the PCO enzymes. So these initial studies validated the reaction that the PCOs catalyze, demonstrated that PCO activity is necessary and sufficient for regulation of Pr0f stability, demonstrated that the rate of PCO activity is sensitive to oxygen availability, and so importantly showed that the PCOs are an oxygen sensing component, are the oxygen sensing component, or okay, to be completely correct, are to date the identified oxygen sensing component of the flood signaling pathway. Okay, so what can we do with this information? Well, there's evidence that stabilizing Pr0f transcription factors can help improve flood tolerance in plants. So we can see that here with Arabidopsis, which has an upregulation of RAP 2.12, which is one of its Pr0f transcription factors. you can see that in those plants which have that upregulated, so more of the earth transcription factor is present, you get a better recovery after submergence compared to those that don't have it. Over here, we've got a barley plant where the final step in the pathway, so the bit that sends the earth protein to the dustbin, that part of the pathway has been knocked out temporarily. So that led to a stabilization of the earth transcription factor, and it's less visually obvious, but you have a better recovery in this barley plant than in this barley plant to the flooding. But most dramatically, we see it over here with the rice that I was showing you before. So sub 1A is an earth transcription factor in rice, and sub 1A is unusual because it is inherently stable. So it is not, for whatever reason that we don't fully understand, but it is not sent for degradation by the proteasome. And we've already seen that having this earth transcription factor present in rice plants confers significant flood tolerance. So what that suggests is that altering this pathway to have stabilized earth transcription factors could have beneficial effects in terms of generating flood tolerant crops, and manipulating PCOs could be a way to achieve that. Interestingly, PCO sequences are highly conserved amongst crops. I won't go into the details, but basically the more red you see, the more identical or similar the regions of the protein are. And so PCOs are highly conserved. They're present in all crops, all plants, everything you look back to, right back to algae. So what that suggests is that if we find a way to manipulate PCOs in, for example, the model plant in Arabidopsis, then we could most likely apply that same mechanism by engineering the same parts of the protein and the same parts of the enzyme, in PCOs from different crops. So our strategy is to manipulate PCO activity in an efficient and targeted way to stabilize the earth transcription factors and improve flood tolerance. So I'll just take a couple of more minutes just to show you where we are with this so far. So what we've managed to do and publish this year is find the structures of two of these enzymes from Arabidopsis. So here we have, this one is ATPCO4. So this is one of the more oxygen sensitive enzymes. And here we have an iron at the active site, and we've got histidine residues which sort of lock the iron in place. And the iron is necessary because that activates the oxygen molecule to oxidize the cysteine residue. We've got a nice open active site cavity, which we suspect has something to do with the protein substrate binding and allowing the earth transcription factor to bind with the cysteine getting right in there to the active site. And we've got an understanding of the amino acids in the active site of the enzyme. So the kind of chemistry that we have close to the active site where the actual cysteine oxidation event is going to take place. So all these amino acids here will be doing something that contributes to the efficient cysteine oxidation. But we don't know what they're doing. So we have to do experiments to find out what the role of all these amino acids is and whether we could manipulate any of them to change PCO activity. So the first thing we've done is to make some variants. So a variant is basically making a version of PCO4 where we swap one amino acid for a different amino acid with different chemical properties. And I'll just draw your attention to two variants which more or less completely knocked out enzyme activity. So H164D, that's replacing this histidine, it is, with an aspartate. So what that does is it changes the iron coordination at the active site. We didn't actually think this would do anything because what we replaced it with, aspartate, is also known to be able to bind iron, but it clearly did have an effect. And we also mutated this residue as spartate, which is a negatively charged residue, and we changed it to something that's quite similar, asparagine, but doesn't have the negative charge. And both of those mutations had a dramatic effect. So the variants that we generated had very little to no activity. And so we took these two variants and we put them into plants. And so we did this with the help of Francesco Licausi, who originally identified these enzymes. And he's got a version of Arabidopsis where he's knocked out 4 of the PCO enzymes. You can't knock them all out because otherwise the plant doesn't survive. But you can knock out 4. And so here we have the knockout and here we have wild type. And what you can then do with the knockout is you can then replace some of the enzymes. So you could replace the sequence for the PCO4 with the wild type PCO4 sequence, and you get a plant that looks very healthy, looks very similar to actually the wild type plant that hasn't had any manipulation. So we've reconstituted the phenotype here by adding the PCO4 wild type back in. But when we added the 2 mutations back in, so our variants H164D and D176N, those variants didn't reconstitute the function of the PCO. So we didn't reconstitute the phenotype. They look pretty poorly, similar to the knockout. And so that shows that the effect that we saw biochemically with our variants here in the test tube were carried over when we took the same variants into plants. And you could even just go back and forth again. So we had a small amount of activity with the D176N, and we saw a slightly better phenotype when that variant was introduced into the knockout plant cells. So what this does is it demonstrates a proof of principle that changes we make to the enzyme biochemically can be implemented into the plant, and we see a similar effect. But of course, there's a long way to go. So what we're doing now is we're looking at a whole host of other mutations around the active site, and we're looking for things that don't impact the phenotype of the plant so dramatically as this. Obviously, we don't, this is not much good. We're not going to get much crop yield if this is the type of mutation that we incorporate into our PCO enzymes. We're looking for something that's going to alter perhaps the oxygen sensitivity so that the window of time where the ERF transcription factors are stable is increased. So they're stable for longer, but we're not completely turning them on permanently, because that leads to a reduction in yield. And we're not completely knocking out PCO activity to do so, because that obviously has a detrimental effect. And we also, as part of this, we also have to understand what else the PCOs are doing in plant cells to be able to make our interventions specific to the manipulation of the ERF transcription factors. So to conclude, hopefully what I've conveyed to you is that reduced oxygen availability and metabolic crisis are a major threat to flooded plants, but plants have over time evolved adaptive mechanisms to help them survive periods of flooding. However, with the effects of climate change coming on, these adaptive mechanisms are not enough. We need to think both about crops which don't have adaptive mechanisms and can't survive being flooded for very long at all. Maize is one example that is really susceptible to flooding. But also even those plants that are adapted to floods. So these rice plants that can survive for 2 weeks underwater, the extent and duration of the flooding that is being seen now in Southeast Asia is so much that being able to survive flooding for 14 days is not enough, because the floods are lasting longer. However, I believe we can give plants a helping hand, and one way of doing that is to have a detailed understanding of flood response pathways to identify strategies for molecular intervention. So effectively, we're doing the end point of this would be gene editing in crops, so editing the genes which encode for PCOs. And overall, I believe that genetically engineered crops have a critical part to play in climate resilient agriculture. Okay, so it just remains for me to thank people involved in the work. So these are the current members of my group, and a couple of key past members of my group in italics who've contributed to this work. So we've got work on PCOs and flood tolerance. We're also interested in PCO evolution, looking back to algal PCOs, and there's a really cool sort of equivalent enzyme to the PCOs in humans, which also has an important role in human hypoxic responses. I'd like to thank collaborators in Oxford. I'd like to thank collaborators elsewhere. I'd like to thank UKRI and the ERC for funding, and I'd like to thank you for your attention. Thank you very much indeed.
Thank you, Emily, so much for such an insightful talk. So we've got a couple of questions from the audience. Let me read out the first one.
Okay.
Let me see. So this one is more of a comment than a question. It says, "The dog is awesome. Please don't tell it after the talk." From Max.
Sorry about the dog.
No, I think she was very popular. If anything,
she started barking at her own reflection in the window. Yeah.
Aww. But I think, if anything, it's more of a shame that we didn't actually get to see her in flesh.
Oh, sorry. I have my professional head on.
All right, let's have an actual question now. Okay, so this one's from Sarah. Do you reckon it is possible to produce flood-resistant crops by traditional selection breeding methods?
I think it's possible in some instances. So, there, if you've got a crop that has a wild relative that is particularly tolerant to being flooded, and you can identify if that tolerance is driven by one protein, then the gene for that protein will be in one particular locus in the genome of that wild relative. And if you can identify that and then put that into the crop, then yes, that's going to work. So there is definitely a place for that in this whole strategy, but I think I don't think that we would be lucky enough to be able to identify such strategies for all the crops that we need to do that for. So I think both the traditional breeding and the genetic engineering need to go hand in hand.
So you think there's probably just too many genetic variables for like all the plants possible out there that we would need to make flood resistant for it to actually just rely purely on previous standard methods used for selective breeding?
I think so. I mean, a geneticist might argue against me and say that traditional breeding would be better, but I think it's slow. So I think if we could, by understanding the fundamental pathways that drive these responses that are conserved in all plants, then we have mechanisms for genetic engineering that we can implement without the traditional breeding.
Okay. Yeah, I guess traditional methods will you need probably more than 1 or 2 generations for it to actually show up the flood resistant genes that you're looking for.
Yeah, and it's a lot of work. I mean, so is this, but I think the two things have to happen at the same time. And there are lots of people doing traditional breeding. So in IRI, for example, at the International Rice Research Institute, there's a lot of breeding of traits going on. So crossing different rice varieties to try and breed better, stronger, more resilient varieties. That's a major part of their work. So it is happening.
Yeah, that sounds pretty cool. Okay, let's get the next question. So this is from Tom. Has CRISPR-Cas gene editing systems allowed you to rapidly test various gene modifications that would enable plants to thrive in flooded areas, such as borrowing genetic tricks from rice and that sort of thing?
Yeah. So the way I see CRISPR, I think CRISPR is going to be the tool that we use to introduce the variants that we engineer into plants. I think what would be a better tool for, well, I don't know, arguably a better tool for us for sort of rapid screening would be a directed evolution type method of looking at different variants at the active site. However, for directed evolution, you need a lot of things in place. So you would, it's not necessarily the be all and end all. So I'm having sort of thought it all through. I think the targeted rational approach is the most likely to lead to useful outcomes given the tools that we have at our disposal at the moment.
Okay. Yeah. I mean, in general, it just sounds like for this sort of thing, you just need a combination of different tools. You can't really rely on one single method to fix the problem.
Yeah.
Okay. Let's have, I think we've got time for one more question. So we can get that one up. Oh, this is an interesting one from Millie, who says, what sort of effect could these flood-resistant plants have on biodiversity in the environment?
Oh, interesting. So I'm just going to have to sort of think this through on the hoof. My natural reaction is to think that it's not going to negatively impact on biodiversity. I suppose what we're not doing, we're not using wild varieties which show climate tolerant traits. We're not using those and introducing those into the sort of wider general agriculture. However, I don't think so we're not increasing biodiversity in that aspect. We're probably maintaining the status quo. Right, okay. So we're maintaining the status quo. I think probably other people need to think about that sounds bad, but biodiversity could be better maintained with different types of strategies. So I would see that as something very different. So you might have pest resilient crops. So there are people who are working on pest resilient crops, and what these do is they allow crops to survive in the presence of certain pests, and that then means you don't have to apply so much pesticide, and that has beneficial effects for biodiversity. So I think what we're doing in terms of flood tolerance is not really touching on any of that, but the same kind of strategies are being used by people in terms of pest tolerance, which would have a positive impact on I would anticipate to have a positive impact on biodiversity. Yeah,
interesting. I never really thought about that much before. It's a good question.
Yeah, so I saw something today, in fact, which it was a real sort of rational and controlled study on the effect of genetically modified aubergine growth in Bangladesh, I think it was. So it was a sort of controlled trial, and they looked at these aubergines that have been genetically modified to have with a gene from a bacteria which has an insecticide in it specific to the pest that is the problem. And they what they saw was basically the amount of pesticide that people were having to apply was significantly reduced, and the yield of the aubergine was significantly higher. Along with that, the health of the people who normally go and spray this pesticide was also significantly better. But the fact that you've managed to genetically engineer this crop that means that you don't need to spray as much pesticide on your crop as presumably or hopefully got positive impacts for the biodiversity on those fields.
Do you think there's a potential risk though that crops that are very that are resistant to sort of pesticides or pests or insects or whatever might end up outcompeting some native plants that might be in the nearby wilderness, and then that could potentially lead to some negative effects? Or do you think that would be quite a negligent effect on the biodiversity?
I think that would be very specific. So if you imagine you're growing your aubergine plant, which has got better pest resistance, I don't think that's going to, unless you've got a neighboring wild aubergine plant, then I don't think it's going to have any impact on plants growing nearby.
Okay. Yeah. So it would have to, it would have to be something really closely related to it, really have a really negative impact on it.
I think so, but we are off my area of expertise.
Sorry. So unfortunately, that's all we've got time for this evening. Thank you so much to everyone who's tuned in and asked questions. And Emily, thank you once again so much for such an interesting talk. It's been an honor to have you. Join us next week, where we will be hosting Akane Kawamura, who will be discussing the topic of epigenetics. Enjoy the rest of your evening, everyone, and stay safe. Good night.